Hello students, welcome to Examstayaree. I am your biology expert and today I am going to start with the second chapter of the unit Reproduction, naming Sexual Reproduction in Flowering Plants. Sexual Reproduction in Flowering Plants. As you know, sexual reproduction needs the formation of male and female gametes from two opposite individuals. And flowers are the reproductive part of the plant. So today we are going to study these following things under the heading Sexual Reproduction in Flowering Plants. So first would be flower, its structure and definition. Second, pre-fertilization events under which we will study 1. Male reproductive part, which consists of stamen, also known as microsporophyll. Second, microsporangium and Microscorogenesis. Third, pollen grains. Then B, female reproductive part, which consists of carpel or pistil, also known as megasporophyll. See, these are the different terms which are being introduced for the first time, so you should be careful. Second, megasporangium plus megasporogenesis. Third, embryo sac. Pollen grains are also known as male gametophyte. Likewise, embryo sac is known as female gametophyte. So, male reproductive part, female reproductive part, then pollination. Since you know that to meet male and female gamete, the pollen grains have to be transferred to the stigma of the flower. Therefore, the pollen grains are transferred with the help of different agents, which we will study in detail. The process is pollination. Then we will study pollen pistil interaction. Have you ever wondered why a particular pollen grain fuses with a particular ovule? When it reaches the stigma, what are the factors which tell the pollen grain? Yes, you are the right one. There is a pollen pistil, this is a natural way. There is a pollen pistil interaction. So once the correct type of pollen grain falls on the same type of flower's stigma, only then the pollen tube starts to be formed. So pollen pistil interaction. Along with this, we will study outbreeding devices and artificial hybridization. So, what are outbreeding devices? You know that cross fertilization is better than self fertilization. Therefore, because cross fertilization brings about variations. So, to encourage cross pollination and cross fertilization and to discourage self fertilization, plants have created certain outbreeding devices which we will study in detail. And then artificial hybridization. You have studied about hybridization, how artificially it is done. This we are going to study in artificial hybridization. After this, we will be studying double fertilization and triple fusion, and post fertilization events, under which we will be studying embryogenesis and endosperm formation, Along with this, we will be studying seed, fruit, apomixis, which is not actually post fertilization event, but another concept. So besides apomixis and polyembryony. So under sexual reproduction of flowering plants, we are going to study all these aspects one by one. So let us start with structure of flower. So in the angiospermic life cycle, there are two main phases. One is sporophytic phase and another is gametophytic. Phase. Sporophytic phase is diploid, and during this period the plants grow vegetatively, means root, stem and leaves grow. Gametophytic is a haploid phase, and in this phase male and female gametophytes develop. Now, these male and female gametophytes are developed within the flower. Flower, नाम सुनते ही भिन्न भिन्न प्रकार के फूल, उसके अलग अलग डिज़ाइंस, उसके अलग अलग रंग, अलग अलग महक के सामने हमारे सामने आ जाते हैं. Flower always had an intimate relationship with the human being from the time immemorial, be it aesthetic, religious, social, cultural, any event. Flower had been a very intimate part of the humankind, and it symbolizes and represents and conveys the message of love, sadness, happiness, mourning, grief, etc. So, what is a flower? For everyone else, flower is a beautiful thing. But for a biologist, flower is the morphological and structural and genetical structure which helps in sexual reproduction. Flower can also be defined as or considered as compressed shoot on which modified compressed shoot on which sepals, petals, stamen and carpel are arranged in 4 whorls. So, because we are saying that flower is a modified compressed shoot, so before you must have seen that one fine morning when you get up, there is a bud on one of the terminal part of the branch. So, suddenly how that bud comes there? Because that part of shoot has a hormone Floragen which is secreted from the leaves. So, there a floral primordium is formed. A floral primordium is formed. This floral primordium develops into inflorescence which bears many floral buds that later on develop into flowers. So, if we see the structure of a flower, you will see that flower rests on a structure known as receptacle or thalamus. And on this thalamus are arranged these parts, sepals, petals, etc. on the thalamus. The outermost word is made up of sepals. On it is present petals, Then it has a female reproductive part known as carpel, which has stigma, style and ovary. Stigma, which is the landing platform for pollen grain, style through which pollen tube moves to the ovary. This is the ovary. Then there are male reproductive parts known as stamen. Which has a terminal anther and a filament. So this is the structure of flower. Now this thalamus has, you can see this stalk with which the flower is attached to the branch of the tree, and it is known as pedicel. So this is a general structure of a flower. Now let us study the male reproductive system completely and then the female reproductive system. So let us study the male reproductive part. And in this, first is stamen. Stamen consists of two parts, as I have already told, anther and filament. Each anther is bilobed and covered with two protective sheath known as theca. Thus, it is diathecous. It can have one theca also, known as monothecous also, but generally angiospermic anthers are diathecous. So the filament is generally attached either to the thalamus or to the petals. If we see the transverse section of this anther, we will get a structure like this, and you will see that each lobe has two chambers. One thing, that the anthers, there is a longitudinal groove run along the anther which separates the two theca. Therefore, each lobe you will see has two pollen chambers, and in all it will have 4 chambers forming a tetrahedral structure at the 4 corners. These are known as microsporangium. Each microsporangium contains many tissues known as sporogenous tissues. It is these tissues which later on develop into pollen grains. Now, the anther is covered with 4 layers. The outer one is known as epidermis. The inner to it is endothecium. then a few middle layers, maybe of 2 to 3 layers, middle layers, and innermost is known as tapetum. Outer epidermis, inside it endothecium, then 2 or 3 middle layers, and the innermost is tapetum. Out of these, the 3 epidermis, endothecium and middle layers, these are protective in nature and helps in dehiscence of anther, while tapetum, it is rich in cytoplasm, has 1 or more nucleus and provides nourishment to the developing microspores. From these microspores, from these sporogenous tissues develop pollen grain. Let us see how. The process is known as microsporogenesis. So what happens is, some of the sporogenous tissues modify and change themselves into themselves into pollen mother cells, also known as PMC in short form. Pollen mother cells. These pollen mother cells are diploid and they undergo meiosis and change into 4 haploid cells or microspores. 4 haploid microspores. These microspores later on change into pollen grains. Now the process of formation of microspores from pollen mother cells is known as microsporogenesis. How the microspores change into pollen grains? After a it undergoes physiological changes, and then the microspore change into pollen grains. As the anther matures, it dehydrates, and the microspores, which are generally arranged in a specific fashion, they also disintegrate. Normally, they are arranged in a tetrahedral fashion. They may be also arranged linearly or in a T-shaped fashion, etc. So they also disintegrate. The anther dehisces, breaks off, the walls breaks off, and the pollen grains are released. Coming to the structure of pollen grains. Pollen grains, also known as I have told you, pollen grains are known as male gametophyte. As the pollen grains mature, you will see, now these pollen grains of different flowers have different shapes, sizes, architectures, etc. Beautiful colors. You can see them under the microscope in your practical classes. But if you see the structure of pollen grain, it is made up of 2 outer layer walls, 2 layer walls. The outer one is known as exine and the internal to it is known as intine. Exine is hard, thick, and is made up of a chemical known as sporopollenin. This is an organic material and is supposed to be the hardest substance because no acids, bases, any chemical can degenerate sporopollenin, and it can also withstand very high temperatures. Because of this, they can be kept as, pollen grains can be kept as fossils also. At different places in exine are apertures, very conspicuous apertures known as germ pores. Germ pores. Through these germ pores emerge out pollen tube. Emerge out pollen tube. While entine is thin and made of pectin and cellulose. Made up of pectin and cellulose. It is a continuous structure, and from entine only a pollen tube is formed which emerges from the exine. Inside these two walls is the plasma membrane, and you can see that there are two cells inside it, a big known as vegetative cell which has a large irregular nucleus and a small generative cell. And a small generative cell. Generally, the vegetative cell has a lot of reserve food material and it nourishes the generative cells which floats into vegetative cell. The generative cell has dense cytoplasm and a spindle shaped nucleus. So normally the pollen grains are shed at this stage which is known as two-celled stage. 60% of pollen grains are released at two-cell stage. Other 40% they are released at three-cell stage. Why? Because there the generative cell undergo mitotic division to form two male gametes. To form two male gametes. Then it is known as three-celled stage. So in this way, I hope you have understood the male reproductive system, where we have studied about the structure of stamen, transverse section of anther, microsporogenesis, and the structure of pollen grains. In my next class, I will take female reproductive system. Thank you.
For more information, download the app now from Google Play Store.